Welcome, 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 one and all, to Cyberhuman Frontiers. We are going to get into something a little bit different from the threads that we've been on over the last few weeks. We're going to talk about something really amazing, and it's so much a part of our everyday lives, all around us, and in all the life on this planet. We maybe take it for granted, but it is truly amazing. We're going to be talking about DNA. Before we get into that, going to get our community guidelines disclaimer out of the way. This video does not claim to give medical, legal, political, or financial advice. We offer it solely for your entertainment. The points of view and purpose here is not to incite fear, violence, harass, or bully, but rather to share opinions and thoughts with other like-minded individuals curious about the subject. That's why you're here. And that's why we're here too. And we're glad you're here. Are you glad we're here? The opinions on this stream belong solely to the person expressing them and are not a representation of the channel Cyberhuman Frontiers. And this video and our channel in general may contain from time to time certain copyrighted works that were not specifically authorized to be used by the copyright holder, but which we believe in good faith are protected by federal law and the fair use doctrine for one or more of the reasons noted in the Copyright Act of 1976. Excuse me. Yes, I'm back. I'm just still getting over a slight cold here. TMC, how are you tonight, sir?
I'm good. How are you?
Great.
Good.
All right. So, what do you have in store for us tonight regarding DNA? Is it about the gigantic amounts of information that it stores? Is it about the fact that it is copyable, it replicates itself, it's flexible, adaptable, it can be edited now with modern technology? It's certainly portable in many ways, whether in a living form of life or whether in vitro. We've all heard about it, but what do you think? What do you think we'd like to dive a little bit further into regarding DNA tonight?
DNA is the new software, middleware, firmware that we understand cell phones, the internet, and the cloud under today's terms. So, if you were to research any of the things I just said, the cloud, modern mobile technology, computers, the internet in general, DNA will be the biologic version of that. And the goal tonight is to get through approximately 60 different documents that will, at a high level, outline DNA's ability to function as software, middleware, firmware, and hardware for what is known as wetware, which is the fusion of the internet into humans.
Yep. Well, where would you like to start?
Well, we'll start at tab number 1. And
okay.
Yeah, there's quite a bit of information. So we're not going to read in detail a bunch of these things. We'll go until you feel compelled to interject. If you go to Cyberhuman Frontiers, I'm hoping that you'll post this spreadsheet in the links going forward. And what you see is a tab with a bunch of raw links. And the tab is DNA computing. That's what we're going to talk about today. Last show was Voice to Skull. A few shows ago was super paramagnetic nanoparticles, iron oxide nanoparticles. And we did a show on DARPA and the nervous system, which we still need to go back to. But this, I think, is appropriate to discuss prior to reviewing what DARPA has been doing as it relates to decoding and encoding biologic neural code through the nervous system and the brain. That being said, DARPA, this document here talks about in vivo nano platforms. In vivo means in the body, in a living biologic host. And just real briefly, what they're talking about is supporting military readiness through sensing technologies and therapeutics that are performed closed loop systems, if you will, in the body, right next to the body. And a few shows ago, we talked about the raw power of the sensor platform that is a modern mobile device that is the near body or off body device that is going to function like a router, so that all of the devices or genetic computational systems that will be grown in the body, they'll be able to communicate with that router, which is today's modern cell phone. They talk about in vivo nano platforms, right? This program specifically seeks to develop new classes of adaptable nanoparticles for persistent, distributed, unobtrusive, physiologic, and environmental sensing, as well as treatment of physiologic abnormalities, illness, and infectious disease. With that, we will move to the next tab. This is another DARPA project to build travel adapter for the human body. This was 2020. The advanced acclimation and protection tool controlled by the warfighter will address the challenges of travel, such as jet lag and diarrhea. Now, these are benign, cute and fluffy. Let's tell the public how fun this is. Language. And so what they talk about here is doing things like restoring wakefulness, right? And normally we get 5-hour energy, coffee, Red Bull, whatever. And these are chemical methods that disrupt downstream sleep patterns and lead to exhaustion. And so what they're saying here is warfighters typically rely on military supplied food, which is logistically burdensome and may lead to warfighters having to consume local food and water that could cause otherwise preventable diseases. So in America, if you travel to Mexico a lot, you know you don't typically drink the tap water. You want bottled water because the treatment procedures there are different than what's in the United States, and therefore the body is not acclimated to consume readily available water. It's just one example. So they talk about being able to, I'll read it right here. The adapter program aims to develop a travel adapter for the human body, an implantable or ingestible bioelectronic carrier that can provide warfighters control over their own physiology. This system will be designed to entrain the sleep cycle, so being able to regulate when you get tired and when you need to be awake, so that you can zero to a new time zone and or recover quickly after doing something like a night mission that obstructs your circadian rhythms. And we'll continue. Cool picture here. Same program, but I want to show you this picture. And they're using traveler's diarrhea here as an example. But what's really interesting is this idea of a cellular factory, biochemicals, and some other foreign material that is in the body. And the combination of these things create the bioelectronics that will interface with something like a cell phone that will give you, as the warfighter, the opportunity to modulate your body so that you can quickly become better ready to perform your mission. We will keep going. The PREPARE program, preemptive expression of protective alleles and response elements. This is a very interesting program.
Did you say protected alleles?
Yeah. Yeah. So again, you have a cool picture here. You have DNA and chromosomes and some type of a bioelectronic interface. It looks like it's controlling and modulating something or other here. And then what you have here is this, what looks like a lipid encapsulated bioelectronic delivery system. And again, this is the public rollout of this kind of a program. Other researchers, particularly the Solari report, highlighted this program, and they were discussing quantum-based medicine. And a real high-level understanding of that is being able to measure the genetics of a healthy organism and record it onto something as archaic as a tape, like what you'd put on a Walkman. And then you can take the sound file, if you will, in analog form, and you can transport that information and play that information through a system that would deliver energy-based therapeutics, typically with light or other forms of electromagnetics. In the Solari report, they discussed microwaves, millimeter waves, light with lasers, and audio. And in some of the examples they provided, these experiments were done back in the 70s and 80s. They were able to transmute genetics from like a duck to a chicken, where you phased energy through the duck into a chicken egg, chicken embryo. And then when that chicken hatched, the chicken had the beak of a duck and the webbed feet of a duck, but the rest of the chicken was the chicken. Another example was taking something like a farm-raised salmon that has been genetically engineered through evolution and through intervention into something that wasn't what a salmon used to be, a fish. And what they did was locate ancient salmon genetics, and they, again, applied phased energy, and they were able to regress the genetics of a modern day salmon back to a prehistoric salmon. And the outcome of that was 2 or 3 times the size of the salmon of today, more aggressive behavior, and on and on and on. So this program is very interesting to me personally. And I think that as more and more comes out about quantum information systems, quantum computing, and the quantum internet, I think medicine is going to follow suit. And we will see something that resembles something like a med bed. Hang on one sec. My computer is not responding well. With that said, we'll move to the next tab. Pandemic Prevention Platform. They have a West Coast and an East Coast Proposers Day back in 2017. Pandemic Prevention Platform, right? 60 days to slow the spread or to stop a pandemic. As you can see, there is an inoculation delivering medicine, genetic information, therapeutics, antibodies, etc., and then this fusion of whatever's going on inside the body. And we'll just leave that where it is. Microns. So this is where we're going to get into some of the software. Machine intelligence from cortical networks. IARPA is the intelligence agency's version of DARPA. And so there is a segmentation of how money is allocated and spent based on the needs of those organizations. DARPA is part of the Department of Defense. IARPA is part of the intelligence apparatus. And so the intelligence apparatus is obviously interested in gathering intelligence. So there are ways to do that. And there are necessities to refine how the information is collected, collated, processed, and ultimately presented to somebody in the intelligence community to make some sense out of it. And so this program is modeling the human brain in order to better allow AI and cloud computing to learn and process information just like the brain. And the brain processes information in a nonlinear way, which means that AI is being modeled to process large amounts of information in a non-linear way, which leads to a random or stochastic type of environment that may on the surface seem chaotic, but once it's refined, if you will, the results that are obtained are basically at the speed of thought. So it says here, Microns seeks to close a performance gap in computation and data analytics by reverse engineering algorithms of the brain.
One of the big goals is to have processing be at a speed that is the speed of thought and faster, even faster.
Yes.
It's a really big deal. If people extrapolate just for a second with that example that you gave regarding the duck and the chicken and the salmon, the ancient salmon versus the newer one, it really is a big deal to see that it's possible to capture that unique life energy signature and transpose it to something else. That opens a whole category of interaction. So, it basically, we're using electronics and computers to store, profile, record, and play back this stuff. This can be done in any of a number of ways. And I'm going to talk straight, directly about humans. I mean, we're talking about being able to capture a person's brain waves and their thought patterns, specific parts of the brain, store them, maybe modify them, and then do a playback back into that brain that changes what you thought, if you know what I meant by that. And this is like, we are there. We really are there. And it's different, but it's very similar to the type of thing that we're talking about when you're capturing that unique life signature of a particular organism. If we've got this type of interface, and if it's at that level of whether it be the signature of the life form in terms of its physical morphology, or whether it be the signature of the life form's thoughts in terms of what's going on in its mind, meaning a human, it's really sort of almost the same technology, a few different interfaces, a few different sensors, and a few different transceivers, but it's the same idea, in my opinion. What do you think?
Yeah, in a nutshell, that's a good generalization. This website is very basic, and it's been around for a while, and it's still online. You can look this up, and it's one of Harvard's pages for an interface they have with government projects. And what you can see here is some links to Obama's BRAIN initiative, the BRAIN project, and IARPA's Brain Mapping Consortium, reverse engineering brain algorithms, and an article here, the US government launches a $100 million Apollo project of the brain. Harvard, the Wyss Institute of Harvard, MIT, and others. And these things down here are very interesting. Behavior analysis, what they call a connectome, which is something with Cas9 barcoding. So CRISPR Cas9 systems that barcode genetics so that they can be organized and collated. Keep in mind, the stuff is in the body. Developmental lineage with Cas9 barcoding, and expression of RNA and proteins, which everything in your body is made up of a type of protein, via this FISQ technology, which is an illumination technology, bioluminescence. So when we think about Cas9 being genetic modification via RNA delivery system, you can apply bioluminescent genetic code and ultimately allow for cells in the body to express bioluminescence. And what you see over here is a brief general diagram of DNA and its ability to monitor and track things like intracellular activity. And then with these markers here, you can map what they're calling here spikes. These are just general terminology so that there is an overall understanding. Templates correct copies, wrong copies, DNA polymerases. We will continue. Another IARPA document is Functional Genomic and Computational Assessment of Threats. This program intends to develop approaches and tools for the screening of nucleic acid sequences, so a sample of genetics, and for the functional annotation and characterization of genes of concern, so tracking genetics and their change over time, with the goal being preventing the accidental or intentional creation of a biological threat. I'm going to read that again. With the goal of preventing the accidental or intentional creation of biological threats. These things have the ability to address important societal challenges, things like food, energy, and medicine. Next generation computational and bioinformatic tools to improve DNA sequence screening and augment biodefense capabilities through the characterization of threats based on function, and to advance the understanding of the relative risks posed by unknown nucleic acid sequences. And we'll continue. Icarus, integrated cognitive neuroscience architectures for understanding sense making.
Sense making.
Refers to the remarkable human ability to detect patterns in data. And this goes back to the other IARPA program. It was the Microns program, I believe, that discussed being able to model artificial intelligence, the hardware, software, and middleware needed to create a synthetic version of the human brain. And so when you listen to the thought leaders, for example, coming out of MIT, they do a great job illustrating the difference between a traditional computer and the brain as a computer, where the brain functions as a generalist, being able to do things like detect patterns. And a traditional computer is simply a raw computational processor. The intelligence and defense organizations globally want both. And so that's why we're going through these documents in order to create a holistic picture at a very high level so that we understand, for your entertainment purposes only, just exactly what and how our money is being spent. This program is to understand and model how humans engage in sense making, both during optimal and suboptimal, which is biased, performance. And we can get into the nuances of what suboptimal or biased is. But what that really is to me is something like your emotions having an interplay with the logic computational function of your brain, so that, perfect example would be you listen to a song and you have a playlist. So you're going to listen to this song a few times over some period of time. And based on whether you listen to the song in the morning, in the afternoon, or at night, if you listen to the song after you get a promotion or you get fired, that song will have a different experience, have a different impact on your psyche, because there is a interplay between what's going on with your logic center and your emotional center simultaneously. And that's what this program is one of many, actually, trying to replicate in silico. SHARP, strengthening human adaptive reasoning and problem solving. Interesting program here. So adaptive reasoning and problem solving, again, human conditions that are necessary for our survival and for our ability to thrive, are increasingly valuable for information-oriented workplaces where inferences from sparse or voluminous or conflicting data must be drawn, validated, and communicated, often under stressful, time-sensitive conditions. So a simple example of this is, and I'm sure this has happened to everybody, you get a text message from somebody. You read this text message. You have some perception or assumption about what is the intent behind the text message. You then speak to the individual later under some preconceived perception, and when in fact what was intended to be sent wasn't what you received because it was a text message and not something that was more complete with information, something like a phone call. That is something like sparse information. Voluminous information would be an overload of information such that the finer nuances that whoever is sending the information out, those finer nuances are lost in the overflow or the flood of information. And then you have something like conflicting data, which is I say something, but I really mean something else. And that, along with the voluminous and the sparse examples, really all boil down to trust. And ironically enough, IARPA has a trust program, which we'll get to in a minute. This IARPA program is the CREATE program, crowdsourcing evidence, argumentation, thinking, and evaluation. Develop tools and methods to design, improve analytic reasoning through the use of crowdsourcing and structured analytic techniques. These new resources empower multidisciplinary collaboration among analysts to provide the intelligence community with accurate, timely, evidence-based analysis. And so what this is in a nutshell is something that I think most of us can get our arms wrapped around. If you think about entities like Facebook or Twitter, the amount of information that is sent out and received over those networks to be able to structure data sets to understand something like political ideology or to understand the direction of an election. Organizations like Facebook and Twitter have the ability not only to measure and monitor, but to potentially inject code, information, propaganda, or whatever to then influence what is the crowdsourced information. And so this program, there's a lot of additional layers when you think about how the intelligence agencies and the military, for that matter, gather intelligence. Once these systems are built, it's a matter of taking other program developments and then applying those toolkits to a platform like this to then be able to do something like crowdsource genetics to understand what's going on at the genetic level across a population, whether it be a city, a state, a country, or the entire world. The TRUST program, tools for recognizing useful signals of trustworthiness. And this gets back to this notion of I have so much information that I can't possibly process all of it. I need a system that will gather the useful information that's relative or necessary to mission success. The tools for recognizing useful signals of trustworthiness seeks to significantly advance the intelligence capabilities to assess who can be trusted under certain conditions and in contexts relevant to the intelligence community, which means if I know a person is not trustworthy, that doesn't mean that I don't necessarily work with them. It means that I need to understand the finer nuances and the dynamics of the individual or group in order to better utilize the asset. They talk about here being able to do this even in the presence of stress or deception. And so another example would be you have special operations going into a country like Afghanistan. And you have the Afghanistan people, if you will, that have had occupation from one country or another for a very long time. Both parties, special operations and the incumbent population, are under stress and function with deception. How does the intelligence community, or the military for that matter, understand what information is relevant and what is necessary to ultimately achieve an objective? That's really what this program does, using computers. And we'll continue. The KRNS program, knowledge representation in neural systems, going back to this notion of creating an artificial version of the human brain. It says here, when making sense of intelligence data, analysts rely on rich repertoires of conceptual knowledge to resolve ambiguities, make inferences, and draw conclusions. Conceptual knowledge refers to knowledge about the general properties of an entity, as well as its relationships to other entities. Understanding how the human brain represents conceptual knowledge is a step towards building new analysis tools that acquire, organize, and wield knowledge with unprecedented proficiency. Moreover, such understanding may lead to the development of novel techniques for training intelligence analysts and linguists. So we are going to get into some DNA stuff now. Molecular information storage programs. They use sequence-controlled polymers. And this is not a synthetic thing. A polymer is simply a single unit with additional single units that are of a similar or different nature. And they're talking about genetics here. So you can look up polymer genetics or DNA polymer, and you can research to your heart's content about what a biological polymer is. There are synthetic polymers as well. So what they're trying to do here is use DNA as the basis for deployable storage technologies that can eventually scale into the exabyte regime and beyond, with reduced physical footprints, power, and cost requirements relative to conventional storage technologies. And this is really interesting where we're going. I want to show you this. This is their Proposers Day document, and we're not going to read too much of it, but I want to leave this up for a second to kind of give you an idea of what the intelligence community consists of. And it's the military and other organizations, the Treasury, Department of Energy, Drug Enforcement Administration, the FBI, Homeland Security, Department of State. So everybody's involved with this stuff. There is a diagram down here that gets into what it is that we're really going to get after here in a minute. So an exabyte, 1 exabyte cold storage data center, Fort Worth, Texas, 750,000 square feet, over 110 acres, 200 megawatts. The media lifetime, 5 years. 10-year operational cost is $1 billion with a B. Where they're going is using DNA. And in this case, they're talking about DNA mixed with silicon. And there are some interesting documents that I found, particularly with Microsoft, that have patented exactly what you're seeing here, but they line it out where they use DNA as a transistor. And there are other documents that I have somewhere on my computer that talk about DNA in vivo functioning as a transistor. So polymers or molecular scale sequences of physical bits, DNA functioning as physical bits, can have a stable lifetime of hundreds of years and an information density that is greater than 10 to the fifth power times higher than that of a conventional storage media, with a raw limit of 1 exabyte per millimeter cubed. So 1 cubic millimeter is the same information storage capacity as, where'd it go?
An exabyte.
Yeah, I want to show this data center here again.
Which is huge.
There it is. 1 cubic millimeter of your DNA can store what a data center of today can store in 1 cubic millimeter. So here's some cool encoding examples. Not that we're going to break them all down, but really what I want to show you here is this merging, if you will, of what we understand as digital information, ones and zeros, and the conversion of it into specific genetic sequences of your A, C, T, and G. They talk about, I don't know if it's in this document or other, but they talk about the error rate of DNA when it copies being exponentially less likely to have errors in it than when you copy one server drive to another after a server crashes. So what they're telling the public is this: data center that costs $1 billion a year can be put into something that's approximately the size of a hotel refrigerator that only costs $10 million per 10-year cycle. So you have $1 billion per 10-year cycle and 750,000 square feet with 200 megawatts of power going to something that fits on a table in a room, less than 200 kilowatt hours, costing $10 million, having a life expectancy of greater than 100 years. And if we go back to the example of a current data farm, you have a media lifetime of 5 years. A server in a rack, one slot in a rack, the life cycle average is about 5 years. And the media lifetime of DNA is greater than 100 years. So that's this document in a nutshell. And we will we'll keep going. This stuff is amazing. DNA, the ultimate data storage solution. The double helix can archive a staggering amount of information in almost inconceivably small volume. So in a world flooded with data, figuring out where and how to store it efficiently and inexpensively becomes a larger problem every day. And this is going to be true going forward. The more things are digitized and put into the cloud, the more data is going to be transferred over the internet. So they're looking at DNA to start archiving information. And if you've been on this roller coaster ride with us for the last few months, where this is going is a proverbial computer system in your body for your entertainment purposes only. You will have a data farm, a server farm in your body. You will have a processor in your body. You will have RAM. You will have field programmable gate arrays, dynamic circuits that are able to be reconfigured on demand, and all the other fun little sensors that a cell phone has. They'll just be grown or inserted into your body. So let's continue. It says, when most people think of DNA, they think of life, not computers. DNA is itself a 4-letter code for passing along information about an organism. DNA molecules are made up of 4 types of bases or nucleotides, each identified by a letter, A, T, G, and C. They are the basis of all DNA code, providing the instruction manual for building every living thing on Earth. DNA synthesis has been widely used in medicine, pharmaceuticals, biofuel development, just to name a few. The technique organizes the base into various arrangements indicated by specific sequences of A, C, G, and T. These bases wrap around in a twisted chain, the familiar double helix, to form a molecule. The arrangement of these letters into sequences create a code that tells an organism how to form. And so they are taking a biologic component that has evolved for billions of years and fusing computer systems with it. Translation software enables efficient DNA data storage. Los Alamos National Laboratory. So major money, direct government affiliation. Software, the adaptive DNA storage code translates data files from what a computer understands into what biology understands. Computer scientist at Los Alamos and principal investigator on this particular project. Makes it similar to language translation, for example, from English to Chinese. They note IARPA here in the MIST program. And the short-term goal of the MIST program is to write 1 terabyte or 1 trillion bytes and to read 10 terabytes within 24 hours for less than $1,000. Other teams are refining the writing or DNA synthesis and retrieval DNA sequencing components of the initiative. DNA offers a promising solution compared to tape, the prevailing method of cold storage, which was a technology dating back to 1951. So this was April 3rd of 2021, not too long ago. We're going to be getting into DNA computation here in a minute. A smear of DNA can hold 10,000 gigabytes of data. This is back in 2020. The US is investing $48 million to turn DNA into living hard drives. I like this picture. Around the world, warehouses the size of several football fields store millions of hard drives worth of data. Every time we send an email, search Google, upload photos to Facebook, or stream a movie on Netflix, which is to say all the time, those hard drives are put to work. Big tech is building more of these sprawling data centers to keep up with the massive growth in data needs. They're generating so much digital data that our current storage systems won't be able to keep up for long. Already, large-scale U.S. data centers cost hundreds of millions of dollars to maintain and account for nearly 2% of the country's electricity consumption, and they're only expected to grow. So, again, more and more on this genetic fusion. Taking cells out to the movie with new CRISPR technology. The Wyss Institute, part of Harvard. I'm going to stop share and make sure that sound is playing. So
good idea.
Bear with me for 1 second. Rather than read this, we'll go straight to this film. It's really remarkable. And again, this is just what they're disclosing to the public.
Pursued almost every way that we can think of to encode information in compact and long-lasting forms, most notably DNA.
What we're aiming to do, in a grand sense, basically, is to encode information into living cells over time. So we want to be able to write new information into the genome. We use the Crispr adaptation system, and so this is a viral defense system that is natively in bacteria, and it acquires usually sequences from viruses. We found that we could hijack the system and have it acquire sequences that we provided. We can use that to then generate these, what we call molecular records or recordings into the genome of a living bacteria. And in order to put constraints on the sequences, we had them contain information, and the information we used was images. So we used static images, and then we used moving images, which we delivered over time to living bacteria. And then we were able to sequence those bacteria and reconstruct the images.
You want to illustrate what its power is, and motion pictures are one of the largest data sets that we have, but it also represents that we're trying to record biological information over time. And so a horse galloping is biological information over time, and is one of the first examples of recording any kind of motion, especially biological motion.
We take all the pixels of an image, we split them up, and then we encode them into DNA. And
we bring those into the cell by a variety of means, CRISPR, Cas1 and 2, store this in a linear array in order of appearance, which is very convenient for us. And then finally, once they're in the array in order, we can then take a population of cells that have had this experience.
We get their genomes out and sequence them, and we just sequence that one locus, the CRISPR array, and from that we can reconstruct the information that we actually put in.
The capacity of a bacterial cell for information storage is limited, and so in a way we're getting ahead of the game by storing information in a bacterial population because the population has more storage. So you could imagine that maybe a bacterium has room for 4 megabytes of information or so before it starts squeezing out the native genome. We're using much less than that, but if you have billions of cells, you can now get millions of gigabytes of information rather than merely megabytes.
I would hope that molecular recorders over time will become a standard thing that is used in all experimental biology. Right now, if you're trying to get information out of cells, you have to either be watching them or disrupt the system, take the data out of the cells. With a molecular recorder, the cell is cataloging its own data. A system can be allowed to progress and develop without an interference by the experimenter, be logging data that whole time, and then you go in and you collect it at the end. So this really changes the way that you would be able to study complex systems in biology.
So we will
Beautiful.
continue. Yeah. DNA from blueprint to blank slate. This is April 23rd, 2021. In the half century since its structure was identified, DNA has become one of the most powerful and versatile tools in biotech. In 2013, the U.S. Congress designated April 25th National DNA Day to commemorate the successful completion of the Human Genome Project that year. This was in 2003, and the discovery of DNA's double helix structure in 1953. Talks about DNA being a valuable tool. The task of sequencing 3.2 billion base pairs in the human genome seemed like a Herculean task until George Church. The guy who spent all that time, if you noticed in the video, lining up the pens vertically and the whiteout and whatever else on the desk. Who knows why, but whatever. Developed the first direct genomic sequencing method. This method was used to generate the first full genome sequence of an organism. He helped initiate the Human Genome Project the same year, capitalizing on this innovation to allow the first draft of the human genome to be completed in 2000. Editing the Book of Life says here, the discovery of CRISPR-Cas9 revolutionized synthetic biology by enabling DNA to be edited quickly and precisely, and it has been used by Church's group to modify the pig genome in 62 places at once. 62 places at once. In an effort to create transplantable organs that are more compatible with humans. These are chimeric organs at this point. Chimeras. chimeras, based on how you want to pronounce it. While many people are rightfully wary of the idea of rewriting genes of humans and animals, synthetic biology today is largely focused on microbial DNA and has the potential to create many useful tools and products. Let's see what this guy has to say.
How we might use nucleic acids to store data. What is data and what is the cloud? This is a photo of the Facebook data center. It's about 1 million square meters, about $1 billion to construct, and it holds all of your data. Yet 90% of your data is never accessed. This is what we have learned from Facebook, from Dropbox, and from a number of studies. By using storage at this rate, by 2025, we'll run into at least
It's all good, except read-write speed is really an issue.
They don't address that specifically in any of these documents. And the read-write speed is going to pick up speed as the quantum information revolution, in quotes, is rolled out. And when you think about quantum networks being instantaneous, where you can have what they call quantum entanglement or spooky action at a distance, if you can remove time from encoding and decoding, uplink, downlink, read, write, however you want to say it, then it won't matter. It'll surpass computational speeds that we understand today as being the best internet speed you can buy. And most of that computation will be done on the body or right next to the body, and it will be processed and packaged in a way that when it is remotely sent or received, it will basically be instantaneous. And that's the real challenge with this kind of research, is being able to find enough from the same organizations or entities in order to circumstantially illustrate just how far advanced all of this technology is. It's been very well diversified, and I'm sure it's been done that way on purpose. And that's where organizations like IARPA, DARPA, the Janus Group, if you don't know what the Janus Group is, I encourage you to look them up. They specialize in fusing technologies together for governments and defense organizations. And it's far beyond what I've been able to research. It's a very interesting group, the Janus Group. So we're going to start looking at computational functions of cells and genetics. And this is a good diagram of what they're trying to create genetically. And it says here how MIT's new biological computer works and what it could do in the future. It says here scientists have been working for years to make cells into computers. It's a logical goal. Cells store information in something roughly approximating memory. They behave due to the strict rules-based expression of programming in response to stimuli. And they can carry out operations with astonishing speed. Each cell contains enough physical complexity to theoretically be quite a powerful computing unit all on its own. But each is also small enough to pack by the millions into tiny physical spaces, with a fully realized ability to program cell behavior as reliably as we do computer behavior. There's no telling what biological computing could accomplish. They talk about being able to create cellular machines that can perform simple computational operations and store, recall, and possess memory. In principle, they provide the sort of control we need to design and build real cellular computers. But they could just revolutionize cell biology long before that future comes about. Mm-hmm. So, I think we'll continue. Safeguarding genes. This is another piece of the larger project, in my view, that allows for funding and for a PR campaign to be rolled out so that people become comfortable with this idea of biologic computation. $65 million in federal program money to make genome editing technologies faster. This is in 2017. Safe Genes program aims to establish a fundamental understanding of how gene editing technologies function, devise means to safely, responsibly, and predictably harness these things, right? Gene editing technologies, for beneficial purposes and to address potential health and security concerns related to their accidental or intentional misuse. DARPA, in this case, plans to invest $65 million in safe genes during the next 4 years. And those 4 years have now passed by. So, this document, if it'll load, that's unfortunate. There we go. Rapid neurogenesis through transcriptional activation in human stem cells. Advances in cellular reprogramming and stem cell differentiation now enable ex vivo study of humans. Neuronal differentiation. It says this program remains challenging to elucidate the underlying regulatory programs because differentiation protocols are laborious and often result in low neuron yields. What are we talking about here? Talking about using stem cells and programming stem cells to make things like neurons or electrogenic or optogenic or other types of genetic variations in the body. Mm-hmm. So they talk about using mRNA to do this, and what they say here is genetic programs involve the rapid transition from stem cells into neurons. So we'll continue. Another cool video from the Wyss Institute, virus-inspired DNA nano devices. So I want to recap here. This is strictly for entertainment purposes.
Cool.
And this is a very high-level understanding of hardware, middleware, and software, what we understand as modern internet and communication computation devices, but for wetware, which is in a biological organism.
I like to tell people that we are trying to make very sophisticated nanotechnology or nanorobots that will be able to detect and treat disease far better than anything that we currently have.
The major challenge for application of DNA nanostructures in health diagnostics and therapeutics is that they're easily destroyed by biological fluids. So the question that we are trying to address is, how can we protect DNA nanostructures from damage? Lipid encapsulated viruses are among nature's most efficient gene delivery vehicles. So therefore, we were motivated to create virus-inspired devices that could overcome these kinds of limitations.
So a nice feature of DNA nanotechnology is that we can make these structures and then we can decorate them very precisely with other molecules. So in this case, we were able to take our DNA nanostructure and then very specifically attach lipid molecules to it. And these act like handles to direct the assembly of a lipid bilayer around the nanostructure. So we can do that and purify the end product, which is the encapsulated nanostructure. We are very excited to see this remarkable similarity between our encapsulated nanostructures and the virus particles. In nature, form and function are tightly related, and it suggested to us that our lipid membranes around the nanostructures might perform the same function as in virus particles to protect them from their environment. Overcoming this instability has been a major challenge in our field. Now that we've been able to overcome that, we hope that we can develop some really exciting applications.
For example, one potential application could be delivery of therapeutics to a tumor. So in this case, one would want to, in general, to have a container that has a high concentration of the therapeutic, let's say that's going to kill off the tumor cells. And the liposome barrier may help to protect that from dispersal, and the particular formulation with the DNA nanostructure might aid in targeting of this bag of drugs to the tumor and not to get lost somewhere else.
So the rollout is obviously for medical therapeutics and diagnostics for chronic issues that didn't exist 70 years ago. And this goes back to problem, reaction, solution, where you have a problem that is created or identified, and then you react to it in order to achieve whatever the final solution may be. Activating genes on demand. New mechanisms for engineering genetic traits governed by multiple genes paves the way for various advances in genomics and regenerative medicine. Let's see what this one says.
Cas9 is an RNA-guided endonuclease that can be directed to any desired genomic location by simply introducing a small guide RNA. In its natural state, Cas9 binds and cleaves DNA, causing mutations to be introduced into the genome in a targeted fashion. However, by simply mutating a few amino acids within the Cas9 protein, Cas9 can be rendered catalytically inactive and will bind to a target region but will not cleave it. One can now decorate Cas9 with any desired accessory regulatory domain. Then, Cas9 will deliver that regulatory activity in a targeted fashion to any part of the genome. Sort of like a genetic Swiss Army knife, where several different functions can be produced by the same tool, just by switching out the accessory component.
In our work, we focus on turning Cas9 into a potent transcriptional activator to allow scientists to target and induce expression of any single gene or set of genes at a robust level. Taking inspiration from nature, we decided to test if we could mimic the way in which natural gene activation occurs within cells, where several activation domains work in concert to activate gene expression.
And in doing so, we're able to find one particular combination, which we term VPR or Viper, that showed a drastic improvement in its ability to activate gene expression as compared to the first generation activators, achieving up to a 300-fold improvement in activity. The ability to regulate gene expression levels through transcriptional activation provides a powerful means to reprogram cellular identity for regenerative medicine and basic science purposes.
And we will continue. A new genome editing strategy offers potential way to control the spread of insect-borne disease and invasive species. And we can think about the Green New Deal and this whole notion of climate change. And we're going to watch a video about gene drives. Gene drives, it's a cassette that uses something like Cas9 to manage population.
Historically, scientists have been able to alter the traits of domesticated plants and animals, but have not been able to do this to wild populations. Here we use mosquitoes as an example to explain why most genome alterations designed by humans don't persist in nature, and how a recently proposed technology could change that. The transgenic mosquito, tinted blue, has an altered gene inserted into one of its chromosomes. When it mates with the wild-type mosquito, each parent contributes one copy of each chromosome to their offspring. Thus, only 50% of offspring will carry the altered gene, while the other half will inherit the wild-type version from both parents. Even if the altered gene doesn't reduce the likelihood of each mosquito surviving and reproducing, it may persist at a low frequency in the ocean of wild mosquitoes, or it might go extinct after several generations of especially unlucky inheritance. This process is what keeps us from altering wild mosquitoes to prevent them from transmitting diseases such as malaria or dengue. A team led by Kevin Esvelt at the Wyss Institute, the Harvard Medical School, and the Harvard School of Public Health has now outlined a way to build gene drives that can improve the odds that almost any altered gene will be inherited, potentially allowing them to spread through even wild populations. The proposal relies on the CRISPR-Cas9 system, a new genome editing technology co-developed by the same researchers at the Wyss. Gene drive mosquitoes carry both the altered gene, the genes for the Cas9 enzyme, and several guide RNAs that tell it where to cut. When passed to offspring, The guide RNAs direct Cas9 to cut the wild-type version of the gene inherited from the wild-type parent. The cell then copies both the altered gene and the drive when it repairs the damage. Because the mosquito now has 2 identical copies, one on each chromosome, all of its offspring will inherit the alteration in the gene drive. This same process will be repeated in subsequent generations, causing the alteration in the gene drive to spread through the population.
So, a key takeaway there, at least for me, was this notion of introducing genetic information that then passes to offspring or your children. This is an interesting document. DNA barcoding. DNA barcoding. There are lots of people that are working on this, and if we were to recall the video that we watched with George Church and others that discussed recording a video into the genetics and then being able to retrieve that video from the offspring of the genetics, in that case, it was, I believe, some type of bacteria. What that gives rise to is this notion of genetic barcoding biology. So it's useful for synthetic biology. It has purposes for synthetic biology so that improvements and refinements can be made. But it's striking the heart of the matter, so to speak, as it relates to something like being able to buy or sell for your entertainment purposes only. DNA data storage, integrated information storage technology for writing large amounts of digital information in DNA using an enzyme-driven, sustainable, low-cost approach. And this is a beautiful image that goes from old school painting, writing, printing, computer bits that we understand, and DNA. And this is the evolution of the recording and conveyance of information of one type or another. We've already watched this video, so we will not watch it again. Crisscross nanoseed detection. Nanotechnology powered infectious disease diagnostics. Enzyme-free DNA nanotechnology for rapid, looks like ultra-sensitive and low-cost detection of infectious disease biomarkers with broad accessibility in point-of-care settings. Multidisciplinary research at the Wyss Institute developed a solution to this challenge by creating an enzyme-free pathogen detection platform that can target specific nucleic acid sequences and proteins. The technology leverages biomolecular nanotechnology to assemble nucleic acid-based nanoseeds that only form in the presence of specific pathogen-derived molecules. That sounds like binary systems. The nanoseeds then trigger downstream signal amplification via the rapid self-assembly of much larger ribbon-like DNA structures that can easily be detected using low-cost readout methods. The method, called crisscross nanoseed detection, is highly specific and ultra-sensitive due to the suppression of non-specific interactions that cause unwanted false positive results. Let's see if there's a cool video here. Planting the seed for DNA nanostructures that grow to the micron scale. And I don't see a video. So these will be on the spreadsheet. I'm hopeful, Indy, that you can add the link and we can talk offline if we want to do a separate sheet for public consumption. It doesn't matter. Up to you. So now we're going to get to a whole bunch of documents that we're not going to read, but they were shocking when I was browsing them earlier. High throughput functional variant screens via in vivo production of single-stranded DNA. And they're talking about introducing a Retron library recombinering, right, RLR, a methodology for high throughput functional screens that surpass the scale and specificity of Cas9 or CRISPR-Cas methods. And so what this is really getting at is a more advanced, more complex, more interactive way to selectively go in and modify DNA. You can search CRISPR systems online to your heart's content, and there's lots of good information out there. That's been around for decades. And this is the newer stuff that I've been able to find that is the next evolution of selective genetic modification. The Rise of Retrons, a new gene editing technique, 2021. Says here, move over CRISPR, a new gene editing technique enables millions of genetic experiments to be performed simultaneously. CRISPR-Cas9 can be programmed to find and cut specific pieces of DNA, but editing the DNA to create desired mutations requires tricking the cell into using a new piece of DNA to repair the break. This bait and switch can be complicated to orchestrate, and it can even be toxic. Because Cas9 often cuts unintended off-target sites as well as, well, it says off-target sites as well. And so they're talking about this new program called Retrons, alternative gene editing techniques called recombinering. Instead, perform the switch by introducing an alternate piece of DNA while a cell is replicating its genome, efficiently creating genetic mutations without breaking DNA. So I'm going to read that again because it's an interesting concept. This technique called recombinering is performed by introducing an alternate piece of DNA while a cell is replicating. That allows for efficient creation of genetic mutations without breaking the DNA. These methods are simple enough that they can be used in many cells at once to create complex pools of mutations for researchers to study. Figuring out what the effects of those mutations are, however, requires that each mutant be isolated, sequenced, and characterized. And that goes back to the barcoding, in my view. A time-consuming and impractical task. So, Harvard and Harvard's medical school created a new gene editing tool called a Retron library recombinering, RLR, that makes this task easier. RLR generates up to millions of mutations simultaneously and barcodes mutant cells so that the entire pool can be screened at once, enabling massive amounts of data to be easily generated and analyzed. The achievement which has been accomplished in bacterial cells, is in a paper. And here's a cool document image out of that paper. Retrons are segments of bacterial DNA that undergo reverse transcription to produce fragments of single-stranded DNA. Retrons' existence have been known for decades, but the functions of this single-strand DNA they produce flummoxed scientists from the 1980s until June of 2020, when it was finally figured out that retron single-strand DNA detects whether a virus has infected a cell, forming part of a bacterial immune system. And they have what they call this retron library, synthetic or natural DNA, a retron plasmid library. This recombinering creates trackable or barcoded edits, and then a test allows for phenotype measurements, genetic sampling, and genetic tracking of intentional mutation. So we will continue. Enzymatic DNA synthesis. Methods from the computer chip industry aid writing and storage of digital data in DNA. Again, they talk about this information capacity and the problem and energy and all that. And they're talking about the solution being using DNA to store information. It says here, while the group's first strategy using this TDT enzyme as an effective tool for DNA synthesis and digital data storage controlled this enzyme's activity with a second enzyme, they show in their new study that this enzyme can be controlled by high energy photons. High energy photons that UV light is composed of. A high level control is essential as these enzymes need to be instructed to add only one single or a short block made of one of the 4 A, T, G, and C nucleotide bases to the growing DNA strand with high precision at each cycle of the DNA synthesis process. So, high energy photons. And high energy photons, we can look at non-ionizing radiation versus ionizing radiation. Ultraviolet light is the cutoff between non-ionizing radiation and ionizing radiation. And the distinction really is a single photon, whether or not it has enough energy in a photon to move an electron out of a shell, a valence shell. And if you want to research multi-photon ionization or photon resonance, multi-photon resonance, what you'll find is that you can use non-ionizing radiation, for example, radio waves, millimeter waves, or infrared light or visible light. And if you focus that energy concisely enough and you apply multiple photons to a particular location, you can get the same effect as a high energy photon. And I'll let you imagine whether or not you think that's possible. But I strongly encourage you to look up multi-photon ionization. And we'll continue here. They're talking about using sheet music, being able to put that into a DNA sequence.
Easily.
Yeah. And store it and then read it and then have it play on something like a computer. And we can digress, I guess, for a second when you think about some of the other stuff we've talked about, being able to access memories and accelerate learning, and this notion of trying to conceptualize consciousness, and what this technology potentially possesses the capacity to do or to disrupt. So
there's really no harm in using DNA for data storage. No harm at all. I don't see that as a problem. I see that as fantastic. The issues are more along the lines of modifying germlines and, of course, deploying something that passes on by way of a gene drive down through generation after generation, ad infinitum, to completely change what the natural world was. That's really scary. I think it's great for data storage.
the other part I have issue with is this idea of privacy. If my memories are coded to be accessed through my genetics, or if my ancestral memories are coded to be accessed through my genetics, this notion of privacy is really a dominant concern for me. So if genetic information can be introduced into wild type populations, which means open air, outside of a laboratory deployment of new genetic information that potentially has the ability to mutate or sever my connection to my ancestors, or to be able to remotely monitor and evaluate how I think or what I think about, that's no different than Twitter, Facebook, Google, Apple, Amazon, and all your telecommunications companies of today accessing your phone and looking at your emails, your contacts, your pictures, turning on your camera and your microphone. Only this stuff is your genetics. And everything and everyone that ever was and potentially ever will be, that is locked away in some coded format in your genes. So
think of this though, in one sense, we're leaving little traces of ourselves everywhere. You know, you leave flakes of skin, hair, and various things behind all over the place. They just fall off of us. So we're already spreading our own unique genetic code all over every place we go anyway. Aren't we? Yes.
Yeah, I mean, we're all related, whether you think so or not. 7% of human genetics are the same throughout every human. And to your point about sharing genetics just through sloughing or through breathing the same air,
is it really that big a deal for my genetic code to be out there, or yours or anybody else's? Because it already is anyway. I guess the deal is that why catalog it all though? You know, I mean, that's taking it to another level for a purpose, some sort of a purpose and intent.
It's for control.
Yeah. And that's what the issue is.
And your genetics or my genetics or anybody's genetics being out there is important, because what I am, the things that I would be most vulnerable to, you may not be, and vice versa. And so if we share genetics, there's great opportunity to strengthen the species. And that's exactly anathema to how the last few years have been. There's been this massive push to convince everybody that everyone else might hurt instead of help. And that's not how our species has evolved. We have evolved to synergistically work with each other at the nano all the way up to the macro. And
there's another interesting little nuance, and it's been the way that nature has been for, well, I mean, I'm just going to say millions of years, that there is kind of an inherent boundary between the intermixing of DNA information from one species to another. That does not happen naturally unless whatever those animals are, or humans, are able to reproduce and produce offspring.
Yes and no.
A horse and a donkey can produce a mule, but the line ends right there. You know what I mean?
Yeah, on a macro scale, I agree with you. On a micro scale, I don't, and simply because if a mosquito bites a dog or a cat and then bites a human, there's genetic transfer there. And who's to say that the genetic information isn't used or identified and managed in some way, even though it's a cross-species contamination? The only thing that we're particularly concerned with and therefore told about is whether or not a cross-species contamination can make you sick. That doesn't mean that a cross-species contamination doesn't do anything else. That discussion isn't had. So I really think there's something to be said for A, C, T, and G being the basic building blocks for everything we understand as life. We're related to the trees. We're related to the bugs and the animals. And to what extent and to how much interface we have with those other organisms here, that discussion isn't had at our level. But if you go back to other cultures like Native Americans, for example, or other nomadic ancient cultures, they were proverbially connected to the earth, where the interplay between birds and animals and humans and plants, that there was another type of a connection there that wasn't necessarily expressed through what we understand as the five senses. And it's anyone who has a pet, a dog, for example, and cats too, for that matter, but I don't know much about cats. I'm not a cat person, but the connection you have with a pet is a very good example that I think from a philosophical perspective sort of illustrates this hypothesis or this theory whereby a dog, for example, will know when you're stressed or when you're happy or what to do without necessarily being told what to do. And you could argue that it's Pavlovian conditioning or operant conditioning or some type of a learned behavior. But when
there's kind of a different thread that I was on. The ancestral man and woman American Indians could both get bitten by mosquitoes and their offspring don't pop out with 6 legs. You know what I mean? And wings. It doesn't happen. So there's another level of, perhaps, I would grant you, perhaps at the genotypic level, there is some integration of other DNA into ours, perhaps some incorporation of it. But at the phenotypic or expression level, where it actually creates the structure, the offspring structure, there's some sort of a boundary there where natural species don't just start changing into other ones just because one of them bit the other one or because their DNA mixed with each other. You know what I'm saying? There is some sort of a kind of a natural boundary. But with gene editing techniques, we're able to actually force that to happen. We're able to force traits from one species, totally unrelated to another one, to be expressed in a different one. That's kind of what I was getting at.
Yeah. What I was getting at was when you see videos today of like a goat and a monkey who are friends, or an elephant, like I saw this today, an elephant, a young elephant for that matter, saw a person in the current of a river, and the elephant intrinsically knew to go help. And
that's cool.
Yeah. And how do you explain that? And I'm not saying it's happening at the genetic level. What I'm getting at is I think there are underlying interconnectivity that's occurring cross species, that when you think about something like a mosquito transmuting or transferring genetic information, it's a way to create common antenna systems so that the human as a transceiver has a remote connection, if you will, to another species transceiver. Everything has DNA. And if you look up DNA as an antenna or DNA as a transceiver, there is a wide variety of information out there that discuss how DNA functions just like a cell phone antenna. And that has absolutely nothing to do with whether or not you get sick. So I agree. There are two different tangents of thought there for sure.
Yeah. There's a lot that we don't know. Some people may know, or may be working on trying to figure it out, but there's a lot that we don't know.
So I'm going to breeze through the last few of these documents simply to illustrate that there is a wide variety of people and organizations that are looking at translating what we understand as modern computer science into biological computational science. This one right here, a new era of semiconductor genetics using ion-sensitive field effect transistors, the gene-sensitive integrated cell. So they're using chemical processes and other processes that occur inside of biology to govern yes and no logic functions like Boolean algebra. There's some feedback coming through, just so you know.
Feedback right now? What is it? I don't hear it.
I do. So this is from NASA. Toward evolvable hardware chips, experiments with programmable transistor arrays. So this is early research published that discusses using transistors and computer systems, but using what they call genetic algorithms. And that's something, there's genetic programming. We'll just search genetic here real quick in this document. So this idea of looking at biology for a number of different things in the silicon world has been around for a very long time. Genetic algorithms as an evolutionary mechanism to configure, in this case, a frequency discriminator from digital gates on available field programmable gate arrays. And you can look those up. Those are in basically every device. Genetic code. Each circuit gets associated a genetic code or a chromosome. Principles of evolutionary synthesis of electronic circuits. And I think we will continue. This is a cool document, but we're running out of time. So this document, Engineering Life, Building a Fab for Biology. Principles and practices learned from engineering successes can help transform biotech from a specialized craft into a mature industry. Although the term genetic engineering has been in use for at least 3 decades and recombinant DNA methods are now mainstays of modern research, most biotechnologists work with living things, or that work with living things has little in common with engineering. So what they talk about here is building these tools to engineer biology. This is a good visual diagram of a printed circuit board with DNA being infused into it. 2006 Scientific American. This is a cool image. Program bacteria. So lawns of engineered E. coli detect chemicals emitted by senders placed in their midst. The receivers then produce different colored fluorescent proteins depending upon their distance from the senders, which they judge by the chemical's concentration. So what this will translate to in biology is something like using bioluminescence that is a gradient bioluminescence that does something like provide remote imaging of internal cell structures, cell membranes, cell scaffolding, and allow that to be imaged with something like a camera. And I say camera, don't limit yourself to thinking that it's a camera that's like a CCD camera where it processes red, green, and blue. Cameras are available in the infrared, and cameras are available in the microwave and millimeter wave. It's simply a reception rather than a transmission of light or photons. And if you can bioluminesce and that be a visual indicator of something, you can extrapolate resonance or other types of illumination outside of a color spectrum, what we understand as visible light. And that could be something as a harmonic or a subharmonic of a cellular structure's resonance that can then be read by an antenna system that works in the millimeter wave or in the microwave. And there's a whole lot more to discuss on that, but it's kind of outside the scope today. If this will load, this is an IBM document that talks about making genetic chips. And let's see if it'll load. Yeah. DNA transistors. Hopes were high for major advances in medicine when the U.S. spent $2.7 billion on the Human Genome Project. 1990, Department of Energy, National Institute of Health. And what you have here is this idea of DNA on a computer chip, which when we go back to that Micron's program where they took the data center and they put it into the size of a refrigerator, this is kind of what that looks like, where you have DNA that is interfacing directly with silicon. And that's something that's not necessarily inside the body. But these companies that are the monolithic platforms have been at the forefront of all of this for a very, very long time. Stanford researchers create genetic transistors, make biologic computing possible. And what you see here is an XOR gate, and it's part of Boolean algebra. And when you look at computer programming with modern transistors, is basically how they understand complex data computation using ones and zeros. And diodes and other components are all part of how a Boolean mathematics system is implemented and what we understand as modern computational systems. And they're doing this with cells at this point, where they're using Boolean algebra to understand yes or no, on or off, 1 or 0, from cellular respiration, cellular activity, from changes in pH to different proteins to different enzymes. All of those things at this point are being studied and or have been studied to then use Boolean algebra to dictate and drive the storage of information, the extraction of information, or the manipulation of the genetics and or the cell or the biologic host as a whole. Systemic design methodology for robust genetic transistors based on input-output specification via promoter RBS libraries. Synthetic gene transistors are vital for signal amplification and switching in genetic circuits. And they talk about having giant repositories of these types of libraries that turn on or turn off some biologic function. And this was an interesting website. This is Harvard, and we're going to highlight some of these. DNA solution of hard computational problems, an autonomous molecular computer for logical control of gene expression, solution of a 20 variable 3-sat problem on a DNA computer, logical computation using algorithmic self-assembly of DNA triple crossover molecules. These are back in 2000, 2002. Programmable and autonomous computation machines made of biomolecules, 2001. Different adiabatic quantum optimization algorithms. And so the quantum component is a piece that I don't understand well enough yet to really drive at these modern telecommunication systems up to and including what we'll understand as 6G, which is this networking of light, being able to use LEDs and pulse modulation with compact fluorescence and all of the infrared laser systems that exist and all of the digital AMO LED and other types of screens that are now on every device. These things can be pulse modulated faster than the eye understands a refresh rate. So to the eye, it looks like a stable picture, but to the subconscious or to the cell or to the DNA, if the light, for example, is able to pass through the body, particularly in infrared, there is an opportunity there to interface with a harmonic or a subharmonic oscillation that then allows for something like light to go in and start modulating, manipulating, reading and writing genetics, organ systems, nervous system control, all of it. DNA machines, experiments, and simulation models of a basic computation element of an autonomous molecular computing system. Autonomous, automatic, self-managed, autonomous molecular computing system. More on quantum, DNA computing, molecular computation, RNA solutions to chess problems. That's interesting. Hardware efficient variational quantum Eugene solver, blah, blah, blah. So let's go to the next website that had some interesting stuff in it. DNA solutions of a hard computational problem. This was one of the documents that we read on the last site that took me to this site that has all these documents. Algorithmic self-assembly of DNA. Simulations of computing by self-assembly, 1998. Simulating Boolean circuits on a DNA computer, 1997. DNA computation, 97. Towards parallel evaluation and learning of Boolean microformulas with molecules, 97. Algorithmic and logic operations with DNA, error-resistant implementation of DNA computations. DNA algorithms for computing the shortest path, DNA computing based on splicing, the existence of universal computers. Programmable chemical controllers made from DNA. Chemical controllers. Chemical controllers. We are electrochemical beings. Humans, electrochemical. Programmable chemical controllers made from DNA. Universal computing by DNA origami robots in a living animal. That sounds fun. Electronics using hybrid molecular and monomolecular devices. Electronics using hybrid molecular and monomolecular devices. DNA computing circuits using libraries of DNA-zyme subunits. So this stuff is big data, catalog, collate, access the cloud, and off we go with directed energy systems for your entertainment purposes only. Autonomous molecular computer for logical control of gene expression. And logical computation using algorithmic self-assembly of DNA triple crossover molecules. Some of this may sound familiar. The documents I was able to find for free, I have, they're here, they're loaded. So this is what you're looking at. And if these links go out, you'll have the ability to archive them yourself. DNA Molecule provides a computing machine with both data and fuel. Unique properties of DNA make it a fundamental building block in the fields of supramolecular chemistry, nanotechnology, nanocircuits, molecular switches, molecular devices, and molecular computing. Autonomous molecular automation. DNA molecules serve as input, output, and software and hardware using ATP as fuel. ATP is necessary for all cellular function. And so this document talks about basically power harvesting your own energy conversion systems and then fusing that with DNA computation functionality so that you have a truly closed loop onboard system inside of your cells or a cell or an organ system or the host in general, using software and hardware made out of your cells. Computing with DNA, the manipulation of DNA to solve mathematical problems in redefining what is meant by computation. Pretty colors. And DNA computing, models and implementations. Talk about this idea of scaling down and where we're at with computing or processing power. 5 nanometers is basically the size with which you're able to bunch a bunch of transistors together on a processor chip before you start smashing up against the limits of physics as it relates to classical computing. And that's why DNA is so useful, because you can build smaller units, nanometer size, smaller than 5 nanometer sized units, and start using things like proton tunneling, which is a quantum mechanical function to perform computation. And as I said earlier, as quantum information science begins to unfurl and reveal what they're going to call the latest breakthroughs in quantum computing, quantum information, quantum medicine, more and more of these types of ideas, I think, are going to come online. And we'll continue. DNA, origami, nano machines. Beyond DNA origami, a look on the bright future of nucleic acid nanotechnology. And they're talking about creating a multitude of 2-dimensional and 3-dimensional scaffolds that form the basis for functional nano devices. Not limited to nucleic acids, these nano devices can incorporate other structural and functional materials such as proteins and nanoparticles. Proteins and nanoparticles, making them broadly useful for current and future applications in emerging fields such as nanomedicine, nanoelectronics, and alternative energy. And this was back in 2011. Document on DNA nanomachines. This is a lab in Japan. There are all kinds of documents here. Japan is at the forefront of a lot of interesting technology. They're very, very, very interesting technology. And some of the stuff is translated in English, and most of it that I found briefly was not. And I was disappointed because I think there's a lot of really good information here. So the next one, molecular computation, RNA solutions to chess problems. And they talk about basically implementing playing a chess game using RNA. And electronics using hybrid molecular and monomolecular devices. And this is the miniaturization piece I was telling you about with the transistors. It says semiconductor industry has seen a remarkable miniaturization trend driven by many scientific and technological innovations. And they talk about it reaching the scale of atoms or molecules. It says a goal that will require conceptually new device structures. The idea that a few molecules or even a single molecule could be embedded between electrodes and perform the basic functions of digital electronics, rectification, amplification, and storage was first put forward in the 1970s. This concept is now realized for individual components, but the economic fabrication of complete circuits at the molecular level remains challenging because of the difficulty of connecting molecules to one another. A possible solution to this problem is monomolecular electronics, in which a single molecule will integrate the elementary functions and interconnections required for computation. DNA computing circuits using, this is a repeat. So this is an interesting one. Computational biologics. And this is probably my favorite picture right here. This really sort of summarizes this notion of a complex biomolecular computational machine. You've got these ones and zeros on the outside of the helix, and then you've got these interconnects that we could say function as a biologic internet that are analogous to what we understand as the cloud. So at this scale, when we look at how genetics or DNA interface with each other, they do so with chemical signaling, they do so with a variety of electromagnetics, biophotons, and they do it with quantum theory, if you will. And what I'm getting at is this: when a baby, for example, goes from 2 cells basically into 4 and then 8 and then 16 and then 32 and then 64, and ultimately you get a baby. The idea of spooky action at a distance or quantum entanglement means that from those first 2 cells, when they fuse and ultimately become 2, 4, 8, 16, 32, there is a connection that exists throughout the life cycle of the human that allows for every single cell to be in constant, instantaneous communication with every other cell. Spooky action at a distance, quantum entanglement in biology. And that is all I got.
Well, that is quite an earful there, TMC.
Yeah. Good thing I have some kombucha here.
Yeah, pretty good. Pretty good. Lots of ways of looking at this. We're going along in the discussion, going parallel with what you've been presenting. There are good arguments to be made to go ahead and enhance ourselves with what technology affords us. Why would we not do that? But it really gets to that fine line of when you've gone over the edge and you're making changes that, I don't know, whoever it is, making changes that they do not know what the ramifications of that are going to be down the road. And secondly, and I guess another argument that could be made would be, if we were, for example, we were just talking about being able to see in other areas of light spectra. If we had utilizing some of the optics and integrating those as an implant into a human and connecting it to the visual cortex, well, that'd be great. You'd be able to see in other wavelengths of light that we can't even see. But the other argument would be, if that was the way that, and you know, we've been developing and growing, and we've been changing and becoming more advanced just by natural processes, if that was where we were supposed to be, is there a reason to wait for nature to cause that to happen, or should we push it? Let's say that 5,000 years from now, the human eye may have developed in such a way that it can see in the infrared or it can see in the ultraviolet. Is it appropriate for us to push that now? And if we have the intelligence to be able to do so, wouldn't we be throwing away that gift of intelligence that we have if we didn't capitalize on it and I guess I would say accelerate our advancement? What do you think? What do you think?
Well, it boils down to one word, which is intent. And if you were to take this idea of intent and apply it to anything that humans do or build, it is the delineating factor between help or hurt. If we are going to realize the human species becoming a type 1 terraforming off-planet exploratory species, then we need to get our arms wrapped around intent so that we fundamentally and intrinsically realize that regardless of what it is we do, we help instead of hurt. If we can't figure it out amongst ourselves, what business do we have looking for other planets or other life elsewhere if all we do is destroy?
Again, it's not about the technology. It just isn't about the technology, as you so well just embodied with your response there. There is nothing inherently wrong with having better vision, better strength, better endurance, better hearing, anything. Nothing inherently wrong with any of that at all. The question is it something that will become or would become available to humanity in general for the purpose of good, or is it something that only certain people will get and others won't, and creating a kind of another kind of a class division between people, the old throwbacks and then the ones that are really advanced, and who gets to be one of them? It's wow. This is just such a complex discussion.
Yeah, if the species doesn't evolve together, we will have 2 or 3 different versions of human that will place
which ones are going to be the most survivable, TMC? Let's say that there are
those
2
that
or
have
3.
the power.
Okay.
Yeah, that's and it goes back to the intent. If people in power look at humans as commodities or as livestock, then you have a split of the species, and that is the opposite direction, in my view, of what I think we're supposed to be doing here. I think we're supposed to be figuring out how to commune with each other rather than feed on each other.
How about this? What about just kind of a general, and I'm just throwing this out, what about just a kind of a general theorem that most of us regular people, I just think of us as regular people, but we're all unique and we all have our areas in which we shine, yes, but let's just call us all the regular people and then call whoever those others are, I don't know, the elite crew, or, the ones who think that they know what's best for us. The kind of people I'm talking about. And I'm not even thinking of anybody in particular. But
White
let's just
shoe
say
boys.
this. What's that?
They're known as white shoe boys.
Okay. White. All right. I never heard that before. That's good.
Yeah.
Let's just say that we came up with just a general theorem, a general rule, which is, okay, so you guys want to build ultraviolet sensing ocular implant. Let's just use that as an example. ultraviolet and infrared sensing ocular implants that are going to be available that can that can really work, and it can be made such that it interfaces with the visual cortex of a human brain. Guess what? You know what? If we can't all have it, then I'm not paying for it. I don't want it. And it is immoral and almost irrational. It's almost antisocial for you to want to spend my money so that you and your pals might be able to have what we will never be able to have. What if we just had a general rule like that? If you're going to build something and you're going to design something with the ingenious, wonderful engineering expertise that you have, and I don't take that away from you, then it's got to be for everybody or it ain't for nobody. What do you think about that one?
Well, philosophically, I can agree with you, but to translate that into a practical environment, that's really the underpinning essence of things like socialism or communism or totalitarianism,
mess,
or fascism. And so it's the real challenge.
But wait, if it's our money that is paying for these, these grants and these programs and whatever the channel of the gravy train of money comes from, if it's coming from us, then we need to be assured that we get what we paid for. Otherwise, we don't pay for it. If those guys want to do it, fine, they can pay for it and they can have it.
You're preaching to the choir, I think, to most of the viewers, simply because we already exist in an environment that functions as some form of socialism or communism or fascism. And it's a perversion of this idea of collectivism. And collectivism, to your point about spending the money and reaping the rewards of the development of that money spent, is this idea of collectivism. But what makes Americanism so unique is you have this idea of rugged individualism. And that is what has been omitted and in fact persecuted by every other form of social governance, is this omission or removal of the individual or of sovereignty. And that's the greatest challenge, I think, for any country at this point, is to establish a meaningful social contract that establishes trust between those that are in charge to make the difficult decisions and those that accept that there are others that are going to make those decisions for better or worse, and will do so operating with integrity and with the fundamental intent of helping instead of hurting.
I feel that we're so far from trust at this point, and I'll bet if I were to take a poll in the group that's watching right now, they'd probably agree. We're a long way away from trusting those who are at the levers of control or funding and all that, to expect that they're going to remember us at the end of the day. I feel as if we're a long way away from having that platform of trust with those people. And I'm not picking on them. I just feel like there's a real disconnect there.
Yeah, if America, the United States, does not correct the core values or the ethos that made this country what it is, then in my view, the species does not have any other avenue to evolve in a way that's going to allow us to survive.
And all right, I hear you. I absorbed what you said. What got me making that initial statement was that we need to simplify this issue, this conundrum, because it's too complex. There are too many variables. There are too many pieces all working together, and very few, I dare say probably none of us have a handle on all of the interconnections and interplay between all of it, although we try our best on this channel to make sense of it. But it's just huge, and we need some simple theorems, some simple solutions, some simple ethical guidelines to go forward by that nobody violates, that is just completely immoral to violate. We need those things, and I don't think that we have them. And it needs to be international. It needs to be kind of like, kind of like just a worldwide understanding. There are just some things that you can do, that you should do, that you should not do, and some that you should never, ever do.
Well, if power were truly restored to the people, then through the ability to fail, to be allowed to fail, through that process, we would establish what is okay and not okay to do to other people, because we would see vicariously, we would learn vicariously, watching other groups making decisions about themselves and for themselves, making incorrect decisions. And without a diversified, deployed sense of sovereignty, where you have an individual having sovereignty and a family having sovereignty and then a community having sovereignty up to the various forms of government, if that goes away, there is no opportunity to have smaller groups of people or smaller entities failing and others watching and observing those failures to then learn without having to go through all of the pain and the mismanagement, if you will, of whatever it is that was used or not used that yielded the failure. And that's why I say if the United States does not reestablish Americanism, there isn't anywhere else in the world that sovereignty at the individual level truly exists. And in my view, to your point about trust with the entities that are in control at this point, man, I totally agree with you. That's really the great challenge of our generation for anybody who's alive today, is to understand the paradigm and then to demand first and foremost sovereignty, but then to be able to inject ideas and thought to allow for spirited debate to occur so that the best ideas can flourish and the species can begin to thrive. And there's no other place in the world that that's even allowed to happen except here in the United States.
Well, I think in one sense, it would be perfectly reasonable for a person to be able to demand not to be invaded by designed synthetic genetic material, which is propagated, right? I think a person should have the right to be able to demand not to be violated, breached, or invaded by such things as that. But let's just talk about this question for a second. Go back to gene drive. The question, to be enhanced or not to be enhanced? That is the question. Modified William Shakespeare. Okay. Well, let's say that there is the opportunity by way of gene drive and deploying a cassette, a sequence of genes throughout the population, which is gradually going to propagate itself through the entire population. And the purpose of that cassette, that design cassette, that gene drive, as it goes down through the offspring and gradually, basically, in a good sense, it infects the entire human race. But the information that it contains is for enhanced hearing or enhanced vision, pick whichever you want, some enhancement. And somebody designed it, and then they let it loose, and it gradually starts propagating, by way of this method through entire populations. Gradually, it in a good way infects the whole world with better vision and better hearing. Now, there's always going to be somebody who doesn't want that because either they're afraid or they want to stay completely au natural, whatever it might be. But how can you make a good argument not to make yourself better in some way?
Well, philosophically, you can't, because we have evolved already through genetic mutation and ancestral experience. So from a philosophy,
I know, but people doing it versus nature, that's the thing. Because now we are able to monkey with nature in very, very powerful ways, right?
Yeah, well, we are people and we are also nature. What makes us unique is that we are basically the only species that the general population understands as being able to manipulate and change our environment in a way,
Right.
a permanent way. So that's the great burden and the great responsibility we have with the planet. And if you take the idea of homesteading, for example, there's a term called husbandry. And what that is this notion that you
Caring for the land.
Right. And the animal and the plants and everything. But
Right.
in the process of that, you're giving life, you're taking life, you're modifying your homestead, for example, up to and including breeding, certain types of pigs or cows or splicing plants in order to improve longevity, sustainability, and all those other cliche terms that have been hijacked by the climate change organizations. And those are very, very difficult challenges. And again, it goes back to intent and ultimately to do no harm.
We aren't the only ones that modify our environment. We can do it very powerfully, but beavers build dams, all right? And birds build nests, all right? So beavers build dams. I mean, how do you think the fish like it when the beavers dam up the stream so that they can make a home in a beaver den for themselves? And the fish can't swim beyond that thing. They might even get eaten by a beaver. So other creatures on this planet do modify their world to some extent, and somehow they work it out. But there is nothing and nobody has ever shown up on this planet that can do it as powerfully as we can. And we're on the verge, we're on the precipice of doing that in some very, I would say, future unpredictability of what some of the outcomes might be.
Yeah, I would agree again, philosophically, but to expound upon that, it's this idea of permanent
Yep.
modification. And that goes
Right.
back to the powerful technologies at this point that we have that allow us at our level to start to begin to see what permanent means, simply because the changes are happening so rapidly at this point. A beaver dam, to your point, isn't necessarily permanent. The trees will rot or wash away or whatever, but
Right.
something like a genetic mutation may never go away unless there's other technology that allows for other types of alteration or regression.
Actually, I think they will. If they do not favor the survivability of the species, talking humans here, then whatever we do, it might take a few millennia, but whatever we do, if it was a mistake, those who got that modification were less survivable will eventually be gone again. Nature will win in the end.
Yeah, the asymmetrical advantage at this point is the dead man switches, the leverage that is implemented such that if they go, we all go. It's a stopgap, if you will, to prevent evolution from weeding out the bad seeds. Again, through the asymmetrical technological advances that the haves have and the have-nots don't.
This is such a complex issue, and it just feels to me like it needs to be simplified. It's just there's so much. This is it's a juggernaut of technology and biomedical technology that's got a momentum of its own. It is not going to be stopped. That I can just my instinct is telling me it's not going to stop. So the question is, who's at the steering wheel?
We either learn or we die. That's it. It's that simple. And that's
some of these esteemed Ivy League speakers that we've had. We've heard a number of little snippets of videos tonight. If they're wrong about what they're working on, well, guess what? It's going to create something toxic, something unsurvivable, something that wipes out a whole block of what the biosphere is. Okay, and in the end, whoever the survivors are the ones who are able to either work around it or adapt to it, and they'll be who's left. And if these people, and I'm not faulting them for their ingeniousness, but if they happen to be wrong, Well, they and a bunch of us might be casualties in the process of it. And I honestly don't know if any of us have the wisdom to be able to look forward enough to see what all the ramifications are going to be.
Yeah, I don't know.
They're playing with this stuff.
Well, that's life. Life is a gamble. Regardless of whether you're dealing with spears and fire to keep away prehistoric animals, or if you're using bug spray to keep, Bill Gates GM mosquitoes off your skin. It's the same thing. It's just the different applications of technology. The intrinsic challenges have really never changed.
Correct. Food for thought.
Mm-hmm.
Well, we've visited with our followers again another week, and we're going to do it again next week. Last words, TMC. Thanking everybody for participating and spread it around. Thinking a little bit about getting into a few more what ifs next week, and a little bit of follow on with some of what the current level of advancement is. I have a few articles and a couple of videos in mind for next week, going a little bit further in this direction, not specifically about DNA, but about just the general convergence of all of this technology in general, and what it means and what there really is going on now. And it's almost hard to believe, but it's really there. It really is there. Some of it's a little scary. And again, it could be fantastic. This is the biggest challenge for humanity right now, is to turn this technological power that we have at our hands toward good, and not toward something that perverts natural order and, the harmony of natural life, both for us and the whole planet. So got some ideas for next week. We'll see what we pull together over the course of the week.
Cool.
So, all right. Well, thanks for your presentation there, TMC. It was stupendous. Thank you, everybody, for participating with us, and we will see you next week. Have a great week, everyone. Good night.